Assalamualaikum, and hi, we are from Group 8 and we're going to present about the effect of salinity and light exposure on marine microbial growth. First of all, my name is Nofarana Alina binti Muhammad Rosli. My metric number is 224797 and I'm going to present about the experimental unit and variable. Experimental unit is the entity that receives a treatment. In this topic, the experimental unit is marine microbial community. For variable, there are two types of variable in this topic, which is independent variable and dependent variable. Independent variable, that something that controlled by the experimenter or something that you make a change in this experiment. The independent variable is salinity, where we measure in part per thousand, and light exposure duration, where we measure in hours per day. And last one is dependent variable, where something that we measure to evaluate the outcome of the changes. So the dependent variable for this topic is marine microbial growth. And I'm gonna pass the presentation to the next person.
Hi, I'm Muhammad Kalimullah Ibadi bin Muhammad Khalidas, and today I will be explaining on completely randomized design, better known as CRD, that we did for this experiment. Now, in this experiment, there are two factors that we are mainly investigating, which are salinity levels as well as light exposure. And in this slide, it is primarily focused on salinity levels, which is measured in the unit of ppt. Now, the CRD is done to ensure that the experiment is conducted in a fair way, and it is a form of randomization. So as we can see, we investigated the levels of between 5 and 35, and including the controls, it makes up a sum total of about 8 different varied treatments. And these treatments are done at a triplicate, which means that it is replicated at about 3 times. Now, in the beginning, before randomization, it is keyed in normally into the Excel sheet from controls all the way until the 35th salinity level or 35 ppt. And then the ran function is keyed in the column beside it, and then it is sorted out. Therefore, that's how we got the arrangement as we can see in the slide, where it has already been randomized and the experiment can be conducted in a fair manner. Now, for light exposure, it will be explained by my colleague Aklan. Thank you.
Thank you, Kalim. And hi, my name is Muhammad Aklam Anwar. And I'll continue on the second factor, which is light exposure duration. For the varied treatment, I included from 0 to 12, and not to forget the control. And for the number of replicates is 3. And as you can see, the results that I get has been randomized and sorted randomly. And in conclusion, for completely randomized design, it is not ideally suitable since we have two different factors with two different units. And later you'll see why CRD is not suitable in this type of experiment. And then I'll pass to the next.
Hi, so now I will walk you through the process of setting up and interpreting a two-level factorial design. It is external design that utilized to study the effect and relationship between multiple factors on dependent variable. It is a simple way to test how two or more factors affect a result by experimenting with each factors at two levels, a low level and high level. In our study of this experiment design, we have two factors. One is salinity and another one is light exposure duration. Since this factor design only allows two levels, we set in the boundaries of 5 and 35 for salinity, and while our light exposure will be set in the boundaries of 0 and 12. Since we have two factors, so we have 4 combinations as stated in table above. This indicates that we have to run 4 times. That's all. Next, I will pass the presentation to Izati. Thank you.
My name is Nur Izati binti Hussein. My matrix number is 223935. Alright, next let's move on to the two-level factorial design. A two-level factorial design is a systematic approach used to investigate the effects of multiple factors on a response variable. In this case, the two factors we are working with are salinity and lax exposure duration, each with two levels, which is low and high. This design allows us to not only examine the individual effects of each factor, but also their potential interaction effects. On this table, as you can see, there is the standard order, which is the predefined order based on the design matrix, and the run order, which randomize these runs to minimize bias. Next, you can see the combination of salinity and lax exposure duration at each run, where levels are represented as either low or high values. Additionally, we included center point represented in run order 6 and 1, which tests the midpoints of the factors level to check for curvature in the response surface. Lastly, we use random formula to help randomize the run sequence and ensure a fair distribution of experiments.
Hi, I'm Hanis and my metric number is 225705. I will be discussing the comparison between two experimental design, CRD and two-level factorial design, both commonly used in research and analyze data systematically, but they differ in their structure and purpose. First, let's define these two designs. CRD is the basic experiment design where treatments are assigned randomly to experimental unit. It is simple and commonly used for single-factor study. Two-level factorial design, on the other hand, is more advanced. It is used to study two or more factors, each of two levels, to explore their individual effect as well as their interaction. For analyzing results, CRD primarily uses one-way ANOVA since it usually focuses on a single factor. Two-level factorial design use two-way or higher-level ANOVA because it involves multiple factor and interaction effect. Next, let's look at factor. CRD typically focus on a single factor. For example, we might test different salinity on the crop yield. In contrast, the two-level factorial design involve multiple factors, such as testing both salinity and light exposure simultaneously, where each factor has two levels like high and low. Both experimental involves randomization. In CRD, treatments are randomly assigned to experimental unit. This helps ensure unbiased results. In the two-level factorial design, randomization applies within the factorial combination to account for interaction between factors. Similarity. Despite their difference, the two assigns share same similarity. Both rely on random allocation to minimize bias. Both involve ANOVA for analyzing results, ensuring statistical rigor. To summarize, CRD is simple, focused on a single factor, and use one-way ANOVA. Meanwhile, two-level factorial design is more complex, study multiple factors at two levels, and analyze both main effect and interaction using two-way ANOVA. That's all.
Thank you, Hanis, for explaining about the comparison between CRD and two-level factorial design. I will continue this presentation with the advantages and disadvantages between CRD and two-level factorial design. Allow me to introduce myself. My name is Nuribiti Zano Arifin with the number matrix 224384. Now, to start off with the advantages of CRD and two-level factorial design. For CRD, it is known as the simplest analysis data design. It also yields maximum degree of freedom. For two-level factorial designs, it has the ability to analyze multiple factors. In this case, we are able to analyze between the lowest and the highest value of the data. It also enables us to identify the relationship between the variables, which is between salinity and the light exposure of marine microbial growth. For the disadvantages of CRT and two-level factorial designs, however, CRD is difficult to find homogeneous experimental units. It is also less accurate to other designs. Therefore, due to this disadvantage, we are unable to use CRD for our to analyze our data. Next, the disadvantage of two-level factorial design is that it requires large sample size, in which we are able to use this two-level factorial design due to our large sample size. And finally, it requires multiple complexity, which means that it requires more than one independent variable for us to use two-level vectorial design. That is all from us. Thank you.